Hi everyone. In this video, we're going to go over the lab, a study of carbohydrates. So we're going to cover each of the sections in this lab. Now the purpose of this lab is to determine how different sugars react with the following. So we have yeast enzymes or fermentation, Benedict's test, Barford's test, Seliwanoff's test, and the aniline acetate test. Now, after we perform all of these tests, we're then going to be able to identify an unknown sugar using our observations from the tests above. Now first, let's go over the different sugars that we're using in this lab. So the first is D-fructose. So notice that in fructose, we have a ketone functional group at the top. So that will be important for some of the tests we perform in this lab. Additionally, there are 1, 2, 3, 4, 5, 6 carbons in this sugar, so that will also be important for some of these tests that we're performing. The other sugar we're going to test is D-glucose, which has an aldehyde at the top. And if we count the carbons, which are just the different points along this Fisher projection, we have 1, 2, 3, 4, 5, 6 carbons as well. D-xylose is another sugar. This one also has an aldehyde at the top, but instead of 6 carbons, xylose has 1, 2, 3, 4, 5 carbons. Now down below that, we have what are called disaccharides. So fructose, glucose, and xylose are monosaccharides, and then down below we have sucrose and lactose, which are disaccharides. So they have two sugars bound together. So as an example, we've got sucrose here, which is one of our sugar samples. And this is a combination of glucose and fructose. And they're connected through something called a glycosidic bond. So sometimes disaccharides will react with some of our reagents differently, or they won't react with them at all, and that will help us distinguish whether we have a monosaccharide or a disaccharide. Our other disaccharide is lactose, and this is a combination of the monosaccharides galactose and glucose. And then our other two samples are gelatin and honey. So going back to fructose, fructose is a sugar that's actually found in honey along with some other sugars. It's also found in fruit. Glucose is a simple sugar that is in syrups. It's also used in other foods as well. Additionally, it's a really important energy source for humans and other living organisms. Now, xylose is a sugar that was first isolated from wood, and it's actually named after that. And then sucrose is basically the sugar we use day to day in your tea or your coffee. Lactose is found in milk. So if you do drink regular milk from cows, that contains lactose. However, if you have a lactose intolerance, then you're probably drinking other types of milk, like almond milk or soy milk. Now, again, honey has a mixture of sugars in it as well as gelatin, so we're just going to test those and see what they look like compared to the other sugars. So let's talk about the first test we're going to do, which is fermentation. So in this test, we're going to react a sugar with the enzymes found in yeast. So off to the side, I have some pictures of yeast as well as bread that's rising due to yeast. And what happens here is that sugar is going to react or ferment with these enzymes. And it's going to turn into ethanol and carbon dioxide. So ethanol has the formula CH3CH2OH, and we're actually going to produce two ethanol molecules, but only one, and two carbon dioxide molecules. But they're only coming from one starting carbohydrate. So in this reaction, we're going to place each of our sugar samples in their own test tube. Then we're going to add a yeast solution. And what you'll probably see are CO2 bubbles coming up from the bottom. And you might also see some foam that forms at the top. So we'll just make some observations during this test. We'll see if there's foam and bubbles. All right. Now the next test is called Benedict's test, and we're going to use something called Benedict's reagent, which is an oxidizing reagent. Now, some sugars are known as reducing sugars. So reducing sugars can be oxidized by Benedict's reagent. So let's write down the general formula here. So a reducing sugar will react with Benedict's reagent, which contains copper 2+ ions. And once we heat those, we'll end up with our oxidized sugar. So it will be a sugar with extra oxygen or extra bonds to oxygen. And then we're also going to end up with a byproduct called copper 1 oxide. So this will be a solid or a precipitate that forms. So if we don't have a reaction with Benedict's reagent, then our solution will actually be blue, which is the color of the copper 2+ ions. So if the copper 2+ ions haven't attached to anything, or they haven't taken an oxygen to form copper 1 oxide, then the solution will remain blue. So that means no reaction. Now, some sugars might turn a greenish yellow color, and that means that we have traces of a reducing sugar. But if you have moderate to large amounts of reducing sugar in your sample, then you're going to see an orange-red precipitate or a brick-red precipitate. And those are the two colors that we'll probably see from our solutions if they do react with Benedict's reagent. So again, in this test, if you do see a reaction occur, then that means you have a reducing sugar. If you don't see a reaction occur, then that means that you don't have a reducing sugar. So what we'll do is we'll perform this test on all of our samples, including the unknown, and we'll compare our unknown to the other results. And that will help us narrow down what the unknown is. In part C, we're going to perform a very similar test to the Benedict's reagent test. This one's called Barfoot's test. So with Barfoot's reagent, we still see an oxidizing reaction occur, but this is a weaker oxidizing agent. So what that means is Barfoot's reagent is only going to oxidize monosaccharides, and it will not oxidize disaccharides. So if you do have a monosaccharide, it will react with Barford's reagent and give us a reddish orange precipitate or solution. If there's no reaction, then the solution will remain blue because Barford's reagent also contains copper 2+ ions, which are blue. Okay, so you can probably already guess, going back to our samples here, we know that fructose, glucose, and xylose are monosaccharides, so those should react with Barford's reagent, but sucrose and lactose will not react with Barford's reagent because they're disaccharides. So we'll just confirm that in our test. Now, gelatin and honey, we're not sure, right, what's in those. So, I mean, you could look up what's in them, but we'll see how they react. And then we'll also compare that to how our unknown reacts with Barford's reagent. So this test is really going to help us narrow down whether we have a monosaccharide or a disaccharide in our unknown solution. All right, part D, Seliwanoff's test. I think I'm pronouncing that right. This test is going to tell us whether we have a ketohexose or not. So a ketohexose would be something like fructose, for instance. So if we go back, remember fructose had 6 carbons and a ketone. So we'll be able to determine if we do have a ketohexose or not. Now, this is also true for disaccharides. So in Seliwanoff's test, you'll see a reaction occur with a disaccharide that contains a ketohexose as well. So, for example, sucrose also contains fructose. So we should see a positive test there, but we'll find out. So a positive test will turn red. So that means you do have a ketohexose. If you don't have a ketohexose, then you'll get a negative test or your solution will just stay colorless. Now in the last test, part E, we're going to perform the aniline acetate test. So the main idea here is that we're mixing our sugar solution with hydrochloric acid, let's see, a aniline acetate, and heat. Now, if you see a bright pink color occur, then that means you have a pentose. Now, a pentose is a sugar that has 5 carbons. Now, if you perform the same reaction and you get a slightly pink color or a dull pink color, that means you have a hexose, so a sugar that has 6 carbons. Now, for this part of the lab, we're going to look at xylose, which we know only has 5 carbons. So we'll see what that looks like. And then we're going to also perform this test on our unknown and see, okay, does it have 5 carbons? So does it produce a bright pink color? Or does it have 6 carbons? So it only produces a slightly pink color. Now, once we've performed all of these tests, we're going to want to write down our results. So when you're watching the lab video, make sure to write down your results in the tables in your lab manual. They look sort of like the one on this slide. So for each one, you could write, let's say like turned pink, or there was no reaction. And then you can compare all of your results with your unknown to try to narrow down what could my unknown be. Is it fructose, glucose, lactose, sucrose, xylose, or gelatin or honey? Now, you could also just use the big table in your lab manual, which has all of the tests listed. So you could say, okay, I did see fermentation with fructose. Maybe you did see a reaction here and here. Maybe you didn't see one there. So you can kind of go through and check off what did react, what didn't react. And then for aniline acetate, we're only going to focus on xylose and our unknown. So there's only 2 tests for that one. Now, a real life example of these types of tests is urinalysis. So if you've ever gone to the doctor's office and given them a urine sample, or if you've had a drug test done for a job, for example, this is one way that you could test your urine to see what's present in it. So you can buy these test strips that you'll dip into urine, and it can tell you based on the color changes whether you have glucose in your urine, if you have ketones in your urine. Let's see, what else? If you have blood in your urine, you can also see what the pH level of your urine is. And typically these strips are used to help you determine if you have an infection, or if it can also be used to test for diabetes. So urine glucose and urine ketones can help you determine if you potentially do have diabetes. Although these strips are not very accurate. So for instance, if you really need to test for urine glucose, it's actually better to use something called gas chromatography mass spectrometry. So it's an instrument that can detect with very high accuracy how much glucose is in your urine, for example. But this is just kind of a quick and easy thing you can do at home to kind of see if you do have one of these things in your urine. All right, so real life example of looking at color changes and kind of ruling out what you have or what you don't have. All right, so that's all we have for this lab. And if you do have any questions about the lab, please feel free to message me.